Today we're going to talk about our committee's progress up till now. If I can have the next slide. As you have seen in the packet, in the pre-meeting packet, the committee has been established since 1996. And the goal of the committee is to provide a forum where scientists from the tripartite nature of this committee, and that is the industry, the government, and academicians, can exchange information and initiate activities which would advance the science related to developmental and reproductive toxicology, which I will refer to from now on as DART. And also to develop strategies to figure out how to deal with the issues that relate to the DART field. Okay, if I can have the next slide. But true to the objectives of the LC and its spirit of tripartite nature, we have had a very successful collaboration between the three parts. And the committee has kept this balance. The chemical industry, the pharmaceutical industry, as well as government from both the U.S. and the European regulators, as well as academicians, have been represented on this committee. The next slide tells us a little bit about who the committee leadership is at this point. I co-chair this DART committee with Dr. James Stewart, who will be talking a little bit later on about some of the projects we're working on. The committee is being led and managed by Dr. Connie Chen, and also with the help of Mr. Oscar Bermudez. We have a number of scientific advisors that come from either regulatory bodies or universities, and also other consultative components as well. The next slide will tell us a little bit about the key project areas where DART has been working on. So as you can see, we put this schema here together for the various aspects of the DART committee work. And this includes the female fertility, male fertility, developmental tox, and juvenile tox. Okay. If I can move to the next slide, there's a little bit more details here about what each circle has in terms of specific projects. Within each category, as you can see, we have been very busy for the past few years developing projects which can be translated into either presentations, workshops, and or publications. A number of these projects will be discussed in a few slides in the following slides to come, but I just wanted to highlight that these presentations have had a wide reach across the globe, and this will be shown on the next slide for global impact reach and impact. I can have the next one, please. So we have had presentations every year in either the U.S., Europe, and or Japan, and these meetings have really represented the various projects that we have worked on. And I have to mention that some of the projects we're working on, we're starting this year to be very involved with other LC-HESI committees, and that include the developmental and immunotox working stream, as well as the germ cell and genetic toxicology projects. Okay, to start with then, I will talk a little bit about one of the projects that we've been very involved in for a few years. This idea came in 2010, so 2010, and the project title is called the the value of the second species. But it's been named many other things, but this is what the name that we refer to it as a shortcut. So this project started in order to figure out whether testing in two species, one rodent and one non-rodent, whether the dual testing provides added value, and also to figure out whether the one or the other species adds more to our understanding of developmental and reproductive toxicity issues. And so the work plan for this project was to collaborate with a bunch of pharmaceutical industry, the members that within the DART committee, where their data, whether it's approved products or non-registered, were pulled from either companies within the U.S. or in Europe. And the data were pulled from rat and rabbit embryonic fetal developmental toxicity study. The data were blinded by the HESI staff, and the committee sponsored a postdoc who was doing the analysis. I can have the second chunk of that pie. And the third one, please. And so what the way this project works is that we use the US EPA data mining program, which is called ToxDRAF database, and the data that I just mentioned from the several companies were put into that database, and then the information was mined from for various questions that we had asked of this database. And so the data analysis has been finished, and if we can go to the next slide, I'll tell you a little bit what the mining has shown. So the results so far, when you look at the analysis by maternal systemic exposure, we saw that for the majority of compounds, there was no meaningful differences in the rat and rabbit maternal systemic exposure at the fetal low adverse effect level. These results suggest that differences between species based on maternal systemic exposure may be often due to stochastic variation rather than being a specific finding. When we did the analysis by, if you can go to the next point, please. When we did the analysis by the severity of the malformation, we saw that in general fetal death was more often observed in the rabbits, whereas the malformations seem to happen more often in the rats. There was a number of analyses that this project was used for, and basically the various analyses modes resulted in no large differences between the two species. The next slide, please.
Excuse me.
So the next, I'm sorry?
Yeah, why do you call that stochastic? Are you truly implying this is random? In other words, are you saying it's technical, or are you saying this is the nature of the biology?
It's more a biological variation, and also we're limited by the data that's in that database, but it's the biological variation.
But stochastic implies that it's random in nature. Do you think it's random?
Okay, well maybe, I have to say we've used the, it's Jane here, these words have been lifted by words provided by some of the people leading this particular work stream. And if you remember, the data is limited by the low, medium, and high dose that the sponsors chose for their studies. So in that sense, it's random. So if the sponsor may or may not be very close to NOEL, and we have to live with whatever doses they chose for their low, medium, and high. So we don't have very extensive dose response data for these animal data. We don't have 9 dose point dose response curves. It's just 3 points.
I suppose this may seem like a minor point to others, but stochastic has a very specific technical definition implying randomness in the system. If this is normal biological variation that makes defining a particular measurement, for example, variation in hormone levels are sometimes often highly variable over different times of the day. That's not really stochastic. It just has a high degree of biological variability based on the nature of the system. The stochastic implies that it's random. I'm just trying to make sure that you're implying there's a good deal of randomness in the system.
Okay.
Jim, this is Tim. It's an excellent question. I think in the interest of time, let's make a note of that, and perhaps we can come back to it at 50 minutes past the hour. But if, Wafa, you would continue on, I think we'll probably be able to stay on time. Jim, hold that question, please.
Okay. All right. So the next steps for this project is that by the end of this year, the two separate analyses will be written in a manuscript, and in 2015, the full story of this project will be published and also presented possibly at SOT and probably at Ecology. And so this project's anticipated impact, basically, there was an ICH meeting last week, and I understand these data and analyses were presented there. What we've learned from this project is very tremendous in terms of actually looking at the studies that have been conducted over the past 10 years. That was the period that we looked at, and to dismiss some myth whether one species is more potent than the other in detecting signals. And so with that, I'll close on the second species, and we can take questions later if it's all right with everybody. The next project that is being wrapped up here is the drugs and biologics in semen. This project was recently presented at a workshop at the FDA. And basically, it was conducted to see whether there is a potential for embryo-fetal harm caused by the presence of drugs and biologics in semen. And so the results that were presented are going to be published soon. And in the future, there might the committee that is working on this project is thinking of a follow-up review where the state of the science for this project will be followed. And so I'll just have this one slide on this project. This project took a lot of collaborative effort and was truly an example of how the various members that are on this committee work together beautifully. Okay, the next project is just starting underway. And this project is about the FcRn ontogeny. And it was created because, as you all might know, there's an increase in developing biopharmaceuticals, which include monoclonal antibodies, where you have diseases that females of childbearing potential might be exposed to. And so this project is being conducted to determine the ontogeny of these concentrations of FcRn in placenta and yolk sac across species, and also to determine the localization of the FcRn in placenta and yolk sac during gestation. At this point, the committee that is working on this project has been assigning each group is going to conduct the analysis on one of the species that are listed on this table. There's a table on this slide. This project will involve actual wet labs where the sources, sample sources have been identified, and the experiments where we have no data will be conducted to put together this map of FcRn ontogeny. And so look forward for the next 3 years to have some results on this project. And so on the next slide, this is a timeline for this project. And so what we're anticipating is this project fulfill a gap in the biology that will apply to placental transfer of all the Fc-containing molecules. And this is mostly for the biologics, basically. And ultimately, this project, if you can scroll down, there are 2 points on the slide that will show up. To improve the ability to extrapolate the embryo-fetal exposure and subsequent observations in non-concurrent species. So this one is to be continued. The next project that I'll mention briefly is the project that we called, and this is Dr. Bob Chafin's credit to the name. So the name Dustin Project is really in honor of the work that Dr. George Dustin has done into the consensus list of developmental toxicity. And as a follow-up, the developmental assay screening for teratology, and we had to finish it with something, so it's with or not. And that's the word Dustin here. It's kind of weird fun, but anyway. So there is a goal for this project, regardless of the name here. The problem statement and objective of this project is to evaluate the compounds and the exposures where you have certain developmental and embryonic fetal toxicity. And then we're going to, the group that is working on this project will use various in vitro assays and to figure out the predictivity of these models compared to in vivo assays. At this point, the next point is the progress report for this project. There has been 10 positive and 10 negative compounds which have been identified. And there are 7 companies which will be working on this project. This is, again, a wet lab kind of project where we're expecting to have at some point the results, and soon, actually, in 2015. And the results would most likely be presented at both a presentation and a publication format. The expected impact of this slide, of this project, is shown on the next slide. And this is to allow people who are conducting in vitro DART assay to evaluate how robust these assays are in terms of positive and negative compounds. And also to compare individual companies or academicians the in-house in vitro DART study to some list that is agreed upon. And what we are hoping is that this list will be a living document in the sense that somebody on this committee or the committee itself will help document all these examples, and so that they will be used as a source of information for the future chemicals that will be put in there. And so look forward also for more results from this in workshops, presentations, posters in the future. And with that, I'm going to turn it over to Jane to take over the next part of the presentation. Thank you very much.
Thank you, Wafa. It's Jane here. Can you hear me all right? Nancy, is this good?
Mm-hmm. Yes, this sounds fine, Jane. Thanks.
Okay, and please
Jane,
go ahead. Thanks. Please go ahead and just fill each slide up. All the remaining slides pretty much fill the page. There you go. So I'm going to start with one of those projects where in the Olympic ring diagram near the beginning, this would be what we describe as a multidisciplinary project. It doesn't fall, strictly speaking, into any one topic. And during our discussions about how we operate and how we use our animal data to inform for human risk, we realized that there was probably quite considerable discrepancy between pharmaceutical companies in how we responded to the presence or absence of animal data regarding what sort of birth control was used in different clinical trial scenarios. And we decided to undertake a survey of ourselves to find out what people were doing to avoid unintentional pregnancy exposure. And so we designed a survey that explored both the methods of birth control, the pregnancy testing, and how the animal data, its presence or absence, and whether it was positive or negative, influenced what happens on clinical trials. And we also asked about what companies did on drug-drug interaction work that allowed women with hormonal methods to join clinical trials. Anyway, the survey went out a couple of years ago. I'm afraid this is my work stream, and so I've still not quite finished the manuscript. It's got quite a long gestation period. But during its gestation, various people were involved with the work stream, including some folks from Europe who we believe went on to influence the European clinical trials facilitation guidance that just came out in September. And what's particularly good about that guidance document is that it clarifies some of the areas of uncertainty that the HESI project has revealed. So we had revealed in our survey that there was uncertainties on how to even define a woman of childbearing potential, and those are pretty important. And so we feel this survey has maybe helped inform certain aspects of the EMA guidance document. And that manuscript is due out, it's due to be submitted hopefully in the first quarter, if not before Christmas. So next, and another point on this is a topic that will be our sponsored symposia at the Teratology Society meeting next June. And one of the issues we will be bringing out is that drug-drug interaction work to support women on hormonal methods, that there isn't very good industry guidance on that. And that is an area of unmet scientific need that needs further work. And we will be trying to draw out some of those things next June at the Teratology meeting. So next slide, please. And just build that, Nancy. So one of the other work plans we have for 2015 is to host a workshop on MicroCT in the spring. And this is the objective of using MicroCT as the example of a modern imaging modality that we would like to be able to use in our developmental toxicity studies, and we'd like to provide the regulatory and scientific framework to make that happen. And so we're hoping that this workshop will bring together the relevant regulators and scientists that will create the framework and the understanding of when we can use these type of techniques. And we feel that this will facilitate the use of other types of imaging modalities, other non-terminal assessments in rodents, and more specifically in non-rodent studies in the future. And one of the things, personally, I feel that preclinical is lagging behind clinical on the use of imaging in its regulatory work. And so it would be good to help bring ourselves up to speed on that. And also that perhaps at times we've had an unhelpful focus on what are probably relative trivia, like the ability to see the degree of ossification in phalanges in using different techniques. These are minor changes that don't truly influence or rarely influence the overarching risk assessments. So we're hoping that workshop will help move things along. So if you go to the next slide, please, you'll see the timeline for this. And so the workshop in next spring and then manuscript thereafter. And we will hope that that will really facilitate the adoption of MicroCT and ultimately other non-terminal and terminal imaging modalities in other types of developmental tox studies. Okay, next slide, please. So the next one that falls into the multidisciplinary category is we are hoping to have a pregnancy and lactational labeling rule workshop possibly in the fall of next year. So we know that the FDA have been writing a rule. It's been out for comment. It's been out for some time. And that the way in which pregnancy labeling is laid out will change fundamentally and be much more focused on the risk summary for the patient and clinical considerations, with animal data having a lesser role in how the information is laid out. So that the way it is laid out will change fundamentally, and the HESI DART group are putting together case studies that we hope will help illustrate different features of the new pregnancy labeling rule once it's out. So we can just go to the next slide, please. So we've been waiting, sitting on the edge of our seats, waiting for this rule to be finalized and published by the FDA, and we hope it's imminent, in which case it will go, the workshops will go ahead next year. And I personally hope that the case materials will be reused by other groups all around the world to help embed this new rule in other territories as well as the US. Because most companies work globally, and so although the rule is for America, individuals sitting in Europe and elsewhere will still have to write these documents in the style required by the FDA. Next slide, please. This next work stream, which is in the topic of developmental toxicity, is a literature review project work stream where the individuals involved are undertaking a comprehensive literature search looking for the data that provides a correlation between exposure to exogenous chemicals and pharmaceutical agents as a means of causing obesity in the next generation. The literature searches have been completed, and I believe, again, I think one of our academic scientific advisors has provided assistance in the form of a PhD of one of his students, and other people are assisting with pulling that data together and the literature together, and a draft manuscript is expected in the first half of next year. And we're hoping that this paper might try and debunk some of the myths that are starting to appear in the literature, as well as casting a light on the data gaps. Next slide, please. So a very specific work stream has been initiated in the DART group to look at potential biomarkers of prolactin concentrations. Now, prolactin in the rodent is notoriously stress responsive and can be elevated by the handling of the animal in the immediate period preceding blood sampling. And so this extreme stress response means it's quite difficult to see treatment effects, or it can be, you can create, you can imply that there's a treatment effect where none is there. And so what this team hopes to do is to find biological biomarkers that respond to a stimulus, the prolactin stimulus, and to perhaps look for things that respond after prolactin has been elevated for a few hours versus has been elevated for a longer period. And that by using these biomarkers of the previous exposure to prolactin, you can then determine whether you have genuinely affected the prolactin responses. Now, this is the sort of work stream that could end up requiring quite a lot of de novo experiments. And so at the moment, it's timetables for maybe 2 or 3 years worth of experiments before they can publish findings. However, if during the data mining phase they can't find any suitable or potential biomarkers that are tractable, then of course this work stream may actually just have a literature phase and move straight to conclusion. So at the moment, they don't know if they actually have tentative biomarkers that might work and be tractable. That's a watch this space. Next slide, please. Now, Wafa mentioned at the beginning that we do have some collaborations with other HESI committees. Over on the right-hand side, you'll see that the Genetic Toxicology Committee and the Immunotoxicology Committee are circled. And Wafa's already described, oh, no, she hasn't. I'll be doing that, take that back. The third one on this list is that we also might have a future liaison with the genomics group to look at things to do with epigenetics. So the genomics committee have just started a work stream looking at epigenetics and using zebrafish as a potential model, and that has been offered to the DART committee to see if it is of interest to our members, and we're just sitting tight to see if anybody bites that zebrafish dangling bait. So next slide, please. So on the immunotoxicology collaboration, there is an ongoing review of developmental immunotox. So there was a paper that came out some years ago, and this is a follow-on activity to address the gaps and see where things have got to since that last literature review was undertaken. And very specifically, this literature review now looks at species that weren't covered in the original articles, so species such as the rabbit and the guinea pig. And it's also looking for the data, the control animal data that might not be available to inform on whether or not some of the methods to look at developmental immunotox have evolved since the previous work stream. And so again, this work stream is hoping to complete their deep dive into the literature next year, and the manuscript would then follow. So that work stream is led by the Immunotox Technical Committee. Then next slide, please. So there is also potential for a collaboration with the Genetic Tox Technical Committee, and this has arisen from following an IWGT workshop in Brazil, where a group of experts were looking at the literature and looking at the world, where we now can see that humans have types of de novo mutations that are causing diseases, and that these de novo mutations are the sorts of mutations that cannot be detected by the traditional regulatory genetic tox testing. And so these have been revealed by whole genome sequencing and other techniques. So things like copy number variants. So all these de novo mutations that arise in humans that could not be detected by the standard techniques we use for genetic tox testing. And the conclusion from this workshop is that we probably ought to go back and reassure ourselves that our current way of operating, where we assume that when we do genetic tox testing on somatic cells, that that is sufficiently sensitive to assure the safety of our germ cells, and that maybe we need to generate more data just to make sure that that is correct. And so that work stream is very much in its early stages, and they'll be developing a work plan in the first quarter of 2015. And the number of DART members who choose to join that is yet to be established. So next slide, please. So this is just illustrating the timelines for all the things we've been talking about. So as you can see down at the bottom, we've got a busy year next year. We've got 2 big workshops, MicroCT and the pregnancy labeling, as well as all these papers to get out. Next slide, please. And just build that. Thanks, Nancy. So we're also committed to presenting and sponsoring various pieces of work all around the world. So the second species work will be presented at least by poster, if not by platform at SOT. We'll be looking at things to do with birth control at teratology. And we may help run a pregnancy labeling thing at the European Teratology Society meeting. So as mentioned previously, a busy time. And I can imagine that in 2016, we may choose to do something to do with pediatrics to assist with ICHS 11. But at the moment, we haven't looked ahead at the workshops for 2016 as a committee. So this slide has been lifted from Sonia Beacon, who kindly lent it to me. And if you just go to the next one, next slide, Nancy. It's trying to illustrate how all these different HESI DART work streams are, in one way or another, helping to influence what we know and understand about our current operating model and how we might operate in the future regards regulatory. So just looking back, in 2009, HESI DART sponsored a maternal tox symposium. It was the first time HESI DART had sponsored a symposium in Europe. We had not undertaken any specific work in Europe up until then. That was the first. And at that, in 2009, we discussed a paper looking at rat and rabbit and sensitivity and influence on agrochemical labeling. And the intellectual discussion then led to the second species project, which is now a very active work stream that is being discussed at the ICHS 5 level. Again, back in 2009, we were discussing enhanced pre- and postnatal development studies and how that impacted the assessment of monoclonal antibodies. And simultaneous with that, we were looking at birth control and how we could keep women who might receive very long half-life monoclonals safe when in clinical trials before we had done any animal work to support those clinical trials. That was a birth control project. And meanwhile, we were also supporting all around the world discussions about alternative methods of assessing developmental toxicity. And you can see all this is helping to set the world in which we're discussing regulatory revisions. So next slide, please. So Tim at the beginning said that this was the 4th time that the Developmental and Reprotox Committee had come for a request for recharter. Well, I hope you're pleased with the progress we've made and have read the document that came alongside this slide set, because the publication list and the presentations we've done, I hope you're impressed, and I hope you ratify us to continue. And last slide, please, Nancy. So here's a list of participants. And also, I just want to say a very big personal thanks to Connie Chen and also to Jim Kim, who was a previous project manager. The HESI DART staff have provided fantastic support to the scientists involved with this committee. So thank you very much to Connie and Oscar, and thanks for your attention.
Well, thank you very much, Jane and Wafa. That was an extremely informative and interesting presentation, I have to say. At this moment in time, what we'd like to do is entertain any questions verbally from the PSSC committee members of a technical nature. If you want to ask Jane or Wafa any particular questions. For those of you not part of the PSSC, if you would just write your questions into the chat box on the right-hand side of the box, right-hand side of the screen. So any questions?
Well, this is Louis Smith, who's a member of the PSSC.
Louis, could you hold on for just 1 second? I think someone else preempted you. Who would that be?
Yeah, this is Peggy Guzipak. I think I have a question for Wafa. I kind of came in a few seconds late. I believe it was her that talked